Hello everyone. Today we talk about advances in genetic engineering of the avian genome, presented by Aliya Gand Mohammed, under supervision of Dr. Istiftah Al-Koum. The main outlines: Introduction, Avian and Biotechnology, Genetic Engineering in Avian, the main method of genetic engineering and gene editing in avian genome, some application in transgenic avian. Finally, conclusion. The introduction. The rapid development of genetic engineering technology, such as transgenic technology and gene editing technology, have provided many great benefits in different areas of biotechnology by using avian species. In this presentation, we discuss different genome modification technology using an avian species. Firstly, we talk briefly about avian and biotechnology, which are a useful model for different fields of research, since the chicken egg is used as bioreactor to produce pharmaceutical and nutrition protein, rather than other mammalian, due to short generation and different phenotypes. Then we talk about genetic engineering in avian. This technology designed to introduce foreign DNA into genome of avian has the potential to enhance current breeding and use for research purposes and commerce applications. Now we show the first approach of genetic engineering is transgenic technology, such as virus-mediated gene transfer. Used in this method, retroviral vector, which generate first transgenic chicken, which inject against to the plastoderm. This vector neutrally integrate to the host chromosome, which lead to form 1 to 11 transformation. Although this percentage it have limitation, such as size of the vector. So has been used lentiviral vector. A transgene is cloned to the lentiviral vector, then preparation transduction to the chick. Then the embryo then cultured to catch and result chimera chicken screened by PCR to identify G0 transgenic birth. The second technique is transposon element. Transposon are genetic elements that can jump between chromosomes. This characteristic of this element makes them useful tool for DNA integration into the host genome. Buggy bag vector. This type of transposon have two interval terms. Repeats flanking encode transpose. It recognizes TTAA sequence in the genome and insert on it. The second approach of genetic engineering is gene editing, such as zinc finger nucleases, TALEN, CRISPR. All types of gene editing depend on DNA repair include non-homologous and joint or homologous recombination. The two ends of the cleavage DNA are joined and ligate, resulting in the generation of insertion or deletion at the site of DSP, according to which mechanism are used. Zinc finger nuclease is the first program in gene editing. A triple of zinc finger is made to bind 9 base pair on the opposite strand. Each zinc finger is fusion to the DNA cutting domain of the FOC1 restriction endonuclease. It has been used in several organisms, such as zebrafish, rat, but has no reports for generation of gene editing in birds. Transcription effector-like nucleases is the second program system, is much more specific for target size, utilized in various species due to easy construction widely and low toxicity. One TALEN is bound to one nucleotide. The most effective and interested in gene editing is the CRISPR. The third program is CRISPR-Cas9. The system is made up of two compounds, include CRISPR, loci, and Cas9 endonuclease, which cleave at the BAM site. Then DNA will ligate to each other or insertion homologous gene to genome. All these techniques are effective in primordial germ cell at stage X, then migrate to dorsal hypoplast. Then gene transfer enter into blood vessel, then enter into gonodial primordial at these stages. Some application in transgenic avian such as egg bioreactor, meat quality, disease model, allergen-free egg. When the application is meat production, in the case of poultry, myostatin is a major target for improving the productivity of livestock, which include the negative regulator of muscle development. Knockout of myostatin significant increase in muscle mass in several animal species. The other application is pharmaceutical protein. In 2018, we produced human interferon in egg white by insertion into ova-albumin gene loci, which generated knock-in chicken. Finally, conclusion. Gene editing tool, which insert into BGC culture, then inject into embryo, then still to hatch and produce transgenic chicken. Then sexual fertilization. Give gene 1 chimera check, which used in many applications. The check, a great model system, becomes even greater. The reference. Thank you for